This is batch 32 from Gokaraju Rangaraju Engineering College, and today we are going to explain you about a major project. The title is Analysis of Biological Microscopic Images Using Segmentation Techniques. In this video, I'm going to go over the objectives and also give you a brief about the whole project. The objective of this project is to detect and segment biological images. The cells in those images after the process are easy to identify, and we can also find the number of cells and area of those cells easily. The whole project is analyzing all the existing pre-processing and segmentation techniques and finding the best out of them. There are three major parts in this project. The first is image acquisition, where we take images from different microscopes, and the second is pre-processing, where we remove unwanted noise and smooth the image for better segmentation. And the third part is segmentation, where we divide the image for better analysis of the image and to get the useful information from it. We are going to use three sets of images to compare the results. To compare the results of pre-processing filters, we are going to use SNR, PSNR, and MSE. And whereas for segmentation techniques, we are going to use accuracy and Jacquard index. There are many microscopics from where we can get our images, such as light field, dark field, phase contrast, fluorescence, and confocal. But we are only going to use confocal and fluorescence in this project. Confocal microscopy is the most advanced microscopy. It converts phase shifts in light passing through a transparent specimen. It has a better resolution, and we can visualize each point of interest separately, so we can get a series of images where we can focus on single cell. It is used for analysis of iconical infections. Fluorescence microscopy depends on the colors, where we use fluorophores to color the cells, and when the cells are colored, we can see them in microscope more easily. It's widely used for clinical and diagnostic process, and it allows to track multiple types of molecules simultaneously because they have different colors in it. These three sets of images are the images we are going to use for the whole project, where we apply various pre-processing and segmentation techniques to it and compare the results. These images are mouse stem cells, HeLa cells, and rat stem cells. After image acquisition from the microscopes of confocal and fluorescence, we are going to apply pre-processing to those images. Pre-processing is a process of filtering an image. It removes any kind of contrast and remove noise. It is done before segmentation so that segmentation can be done properly. And we are going to evaluate the pre-processing techniques using SNR, PSNR, MSE, and RMS. These are the various pre-processing techniques we are going to use in a project, which are mean filter, median filter, Gaussian filter, Wiener filter, and bilateral filter. We are going to apply all these filters to the set of images and compare the results using matrices. Mean filter. It's a simple smoothing filter used to remove noise. It reduces the intensity variation between all the adjacent pixels. This is the MATLAB command for the mean filter, a special IM filter. We are going to apply this commands for processing of mean filter in the MATLAB code. The next filter is the median filter. It is a non-linear filtering technique. It is used to remove noise but also preserve the edge. This filter is generally used in signal processing. This is the MATLAB command for the median filter. Gaussian filter. Gaussian filter is used to remove the Gaussian noise. Gaussian noise arises during capturing of images. Gaussian filter is a non-linear low-pass filter, which gives more weight to the central pixels and less weight to the neighbors, in which and this filter is used for smoothing of images. This is the formula for Gaussian filtering, and we use a 5x5 kernel for the filtering of the image. This is the MATLAB command for applying Gaussian filter to an image. Wiener filter or least mean square error filter. This is a restoration technique. When an image is blurred and has noise, we can restore the image by using Wiener filter. It removes the relative noise and also removes blurring simultaneously. And the MATLAB command for this is Wiener2. Bilateral filter. Bilateral filter is a nonlinear smoothing filter which removes noise and also preserves edges. But for the weighted average of this, we just don't include Euclidean distance but also intensity and depth, which gives us more accuracy. And the MATLAB code for bilateral filter is IM bilateral filter. After applying all those pre-processing techniques, we are going to compare them using matrices. The different matrices we are going to use are SNR, MSE, PSNR, and RMS. These are the formulas for the matrices. After applying the matrices to the LSL, we can see the results here. And we are going to consider SNR and PSNR mostly. And we can see that mean and Gaussian both has the most SNR and PSNR values. Coming to mouse cells, we can see that median plus Gaussian and histogram equalization in median has the most SNR and PSNR values. In rat cells, median plus Gaussian and histogram equalization plus Gaussian has the most SNR and PSNR values. After comparing all those matrices, we can conclude that for the high PSNR values that are mean filtering and median filtering followed by Gaussian, and for the high SNR, we could derive that high histogram equalization followed by median and histogram equalization followed by Gaussian are used. The third step is segmentation. Segmentation is process of grouping of pixels of similar attributes and getting information from it. These are the types of segmentations: morphological, Otsu, splitting and merging, fuzzy C-means, threshold followed by watershed, and morphological followed by watershed. Morphological segmentation is a mathematical form of segmentation. There may be many defects in a binary image, so we use morphological segmentation to avoid those defects and remove noise and get the most important information from those images. Otsu method is used to do operation on clustering based images, and it's also used for reduction of grayscale images into binary images. Splitting and merging is based on dividing the images into a square region of size m by m and then analyzing each square if they have similar attributes, merging them, or if not, splitting them. Fuzzy C-means. Fuzzy C-means is a method of clustering a piece of image or data, and minimization is the main objective of this method. It represents the means of membership algorithms. Watershed is one of the most used human segmentation techniques. It is used to overcome the overlapping problems, and it is used to prevent overlapping of cells in our images. Threshold is a method of changing a grayscale image into a binary image, where we set a value called threshold value. There are three types of thresholding: global, variable, and multiple. We can also apply cascading, where we can apply the output of one technique as input to the other technique. We are going to use two cascading techniques: threshold followed by watershed and morphological followed by watershed. The matrices used to compare all the segmentation techniques are accuracy in the card index. Using these two matrices, we can compare the segmentation techniques and choose the best out of them. After applying matrices to all the segmentation techniques, we can see for the HeLa cells, fuzzy C means and threshold plus watershed gives the most highest accuracy. For mouse cells, we get the highest Jaccard index to threshold plus watershed, and for accuracy, we get threshold plus watershed and fuzzy C means. For rat cells, for Jaccard index, we are going to get the highest values for threshold plus watershed, and for accuracy, we are going to get for splitting and merging and threshold plus watershed. This is the image through the whole project, where we take the original image and pre-process it and apply segmentation techniques and then outline it so we can count the cells easily. These are the image of the mouse cells, original to pre-processing and then applying segmentation, then outlining for them for counting of the cells. These are the rat cells, where the original image is pre-processed and then segmented and then outlined for calculating and counting the number of cells. After applying various pre-processing and segmentation techniques and applying the matrices respective to them, we can conclude that morphological followed by watershed and threshold followed by watershed has the most accuracy and Jaccard index. And after comparing all those matrices, we could easily calculate the area of cell and number of cells.